Welcome. We will start with some basic questions and then go in detail. First question: what is forced degradation or stress testing in pharmaceutical industry? Forced degradation, also known as stress testing, is a process used in a pharmaceutical development to intentionally degrade a drug substance or product under controlled conditions. What is reason behind doing forced degradation study? There are majorly four reasons for doing forced degradation study in pharma industry. These are: first, to identify potential degradation pathways; second, to determine stability of product; third, to develop stability indicating methods; and fourth, to support regulatory submissions. Reference: ICH Q1AR2 section 2.2. Basic but important: what are the common stress conditions used for forced degradation studies? First, temperature; second, humidity; third, light; fourth, oxidation; and fifth, pH. These are the five important stress conditions that are used in forced degradation studies. Reference: ICH Q1AR2 section 2.1.3. What is comprehensive forced degradation study plan? Below are the seven major steps in comprehensive forced degradation study plan. These are: step 1, objective setting; step 2, sample preparation; step 3, identifying stress conditions; step 4, sampling; step 5, analytical testing; step 6, data analysis; and seventh final step, reporting. Next question: can forced degradation studies be used to predict the shelf life of a drug product? Direct answer is yes. Forced degradation studies can provide valuable insights into the degradation behavior of a drug product, which can be used to predict its shelf life. What outcomes forced degradation study offers? Below are the four major outcomes that forced degradation study offers. These are: determination of likely degradants, determination of degradation pathways, determination of intrinsic stability of the drug molecule, and determination of validated stability indicating analytical methods. When and why are forced degradation studies repeated during a product's life cycle? Forced degradation studies may be repeated if there are significant changes in formulation, manufacturing process change, or packaging material change to ensure the stability profile remains consistent. Reference guideline: ICH Q1E section 2.1. What is difference between forced degradation and accelerated stability studies? You can refer below table for understanding the difference between these two terms. The major difference is in their objectives. Stress testing or forced degradation evaluates the intrinsic stability and identifies degradation pathways, whereas accelerated stability studies predict product shelf life and storage conditions of the product. Basic but important: why product degradation should be avoided in the pharmaceutical industry. Product degradation can lead to the formation of unwanted chemicals or degradation products that can affect the drug's efficacy. These products can be toxic or cause unwanted side effects which can pose serious health risks to the patients. Is it mandatory to perform forced degradation studies? Direct answer is yes. International Conference on Harmonization, that is ICH guidelines, make it essential to organize the forced degradation studies, and it is evidently mandated to perform forced degradation of new drug products. When to perform forced degradation studies? According to FDA's guidance, phase 3 of the regulatory submission process is the prominent time for these studies. According to ICH Q1AR2 section 2.2, forced degradation data should be submitted to establish stability profile. What are the experimental conditions, storage conditions, and sampling time for hydrolysis forced degradation studies? Experimental conditions for hydrolysis forced degradation studies are: control API zero point 0.1 molar HCl, zero point 0.1 molar NaOH, acid control, base control, and at pH 2, 4, 6, and 8. Storage conditions recommended are mainly 40 degrees Celsius and 60 degrees Celsius. Sampling time: 1 day, 3 days, and 5 days. What are the experimental conditions, storage conditions, and sampling time for oxidation forced degradation studies? Experimental conditions are mainly 3% H2O2, that is hydrogen peroxide, peroxide control, AIBN, AIBN control, and control. Storage conditions are 25 degrees Celsius and 60 degrees Celsius. Sampling time 1 day, 3 days, and 5 days. What are the experimental conditions, storage conditions, and sampling time for photolytic and thermal force degradation studies? Refer below table for this information. What are the threshold limits for degradation impurities as per ICH-Q3BR2? Refer below table for different threshold type, their description and limit as per ICH-Q3BR2 guideline. The important three threshold types are reporting threshold, identification threshold, and qualification threshold. Which advanced softwares are used for forced degradation study? Below are the four important advanced softwares that are used in pharma industry for forced degradation study. These are Empowers by Waters, OpenLab by Agilent, Chromeleon by Thermo Fisher, and MassLinks by Waters. What is the role of excipients in controlling product degradation? Refer below table for excipient category, role in controlling degradation, and their examples. Antioxidants are used to prevent oxidation. Buffers are used to maintain optimal pH. Chelating agents binds with metal ions to reduce catalytic oxidation. Moisture scavengers are absorbing moisture to prevent hydrolytic degradation, and coating agents are used to create a barrier to protect from light, moisture, and oxygen. What are the advanced techniques to detect product degradation? Chromatography, spectroscopy, mass spectrometry, thermal analysis, and microscopy are the advanced techniques to detect product degradation. Next, very important question: how much product degradation is acceptable in forced degradation studies? In forced degradation studies, the degradation level is generally expected to be within 5 to 10% of the active pharmaceutical ingredient, that is API, under stress conditions. Guidelines reference ICH Q1A-R2. What is impact of product degradation? Reduced potency, toxic byproducts, altered appearance, and regulatory non-compliance are the impact of product degradation. Last and very important question: which guidelines are commonly referred for forced degradation studies in pharmaceutical industry? ICH Q1A-R2, ICH Q1B, ICH Q2R1, FDA's guidance 2018, and USP chapter 1 to 25 are the majorly referred guidelines for forced degradation study in pharma industry. Don't forget to like, comment, and share. Subscribe to channel for more videos related to our pharmaceutical industry.